Thank you. All right, so welcome everyone to Lake Water Quality, Cyanobacteria and eDNA Research with Amanda McQuaid and Jill Emerson. And we are just honored to have these two knowledgeable people with us tonight. So thank you for coming, Amanda and Jill. Amanda McQuaid, who will speak first, is a state specialist in water quality and ecotoxicology for the Cooperative Extension, director of the Lake Slay Monitoring Program, an extension associate professor of limnology and joint faculty in the Department of Biological Sciences at the University of New Hampshire. Amanda also co-manages the Center for Freshwater Biology, our participatory science and research-based collaboration in support of freshwater research, toxic cyanobacteria, and water quality of lakes. Amanda has focused her research on the bioaccumulation of toxins in the aquatic environment, specifically how cyanotoxins transfer through water, air, and food webs. Okay, so we're going to turn it over to Amanda at this point.
Okay, thank you for the introduction. That was a lot. Thanks for having me tonight. I'm very happy to share the stage with Jill, and I just frantically tried to delete a lot of slides because I can go on a lot about cyanobacteria in a way that is probably not normal. Anyway, I want to talk about water quality, but my expertise is cyanobacteria and lakes, lake ecology. In the Lake Slay Monitoring Program, and like Green Mountain and other groups like VLAB across the state, and groups like Chocorua are monitoring lakes, and that's really great. Once in a while, those people who are really carefully looking might find something unsightly or anomalous or just something strange. And so I usually get some calls related to those sightings. When I formerly worked for New Hampshire DES, I was chasing blooms all over the state. But we know a lot more about cyanobacteria as we've gone through the literature and the research. But what we've once thought of photosynthetic bacteria that we would be calling blue-green algae, we still kind of do call this blue-green algae at times. So still look for those in your supplements and in your over-the-counter products. In fact, ice cream dyes. We're talking about, getting rid of whatever red number 5. Well, cyanobacteria is another kind of coloring agent. So there's a lot of things out there. I want to kind of keep this in context to the relativity of all the other terrible stuff there is. Cyanobacteria really not all that bad, but we're seeing a lot more of it, and in some of our big clean lakes that we would not normally see stuff like this in. So we're concerned, but we want to monitor. But cyanobacteria are not new. They've been around for 3.5 billion years. They probably helped us with some oxygen on the planet. They're really not all that bad, and they're not new. They're ubiquitous in the environment. They're everywhere. They're not just in lakes. They're quite diverse. So there's thousands of species and hundreds of toxins, which get us a little upset, is the toxicity and the unsightly nature of knowing that water quality is deteriorating by the site. We can see it. We do focus a lot of our attention on surface blooms because we can see it, but we know that there's all different forms of cyanobacteria, some that live on the bottom of the lake, some that are too tiny to even see, some that are very important for the food web, and some that are not toxic at all. In fact, probably a lot at times are not toxic. But they're still pretty unsightly. And what we're concerned about is climate change and the things that we're doing to the environment to contribute to runoff, that allow nutrients and other things to get into the lake, honestly. So this is more about protecting watersheds, water quality, but it is a sign that there could be some trouble with nutrients and the balance of the ecosystem. Because blooms are just a rapid growth of an acceleration of anything. Algal blooms can be a mix of a lot of different things, not just cyanobacteria. And we hear a lot about red tide and blooms in the ocean. This is, the freshwater version of that. But inland, we are seeing lots of effects on our lakes. Eutrophication, global climate change are major issues. So it's really not just the driving of cyanobacteria, but it's the deterioration of our water quality in general. There's a lot of things to be concerned about with our fresh water, because fresh water is life. Here we are at Chocorua Lake, just such a beautiful setting. You don't see all of that immediate effect of land use in a setting like this, which is great. Nice blue setting. So does that mean that we don't worry about it? Um, every lake is so unique, and we know that watershed influences are changing our water quality rapidly. Of course, again, weather effects are also creating just, sometimes the perfect storm, if you will, to create blooms. There are not only bottom up effects with nutrients, and that's a big part of the equation, but there's also food web dynamics that we should consider in our lake. So every lake really truly is unique in its own way. And therefore, I think it needs to be monitored in its own way as well. We do have a lot of, you know, routine ways of monitoring, but how do we monitor blooms? When we see blooms, they're actually kind of on their way out. They've already been living in the lake. They just happen to be there at the beach when you're there, and that's not fun. So typically not a great sight to go to the beach and see scums. But that's not really probably where they even came from. They get caught in the surface tension, they blow around with the wind, the currents, and weather can have a big effect on that. In fact, even time of day, some mornings it might be nice and calm, and you could see an accumulation coming up to the surface. And then by afternoon, wind fetch might push it back down. Does that mean it's not there anymore? No, it just means that it's moving around. But these organisms are also different, and they do go through different cycles. So think of them as different as plants are from each other. Under the microscope, they just look like these little lines and dots and these blobs, if you will. But there's a whole diversity of different types. And so um, we are looking at methods that Jill will talk about later too, like PCR and genomics, to really understand what we are finding in our lakes. But just by identifying them under the microscope, we can start to group them in these different morphological units, and we can identify them based off of how common they are. So the most common one that we are seeing in New Hampshire typically is this Dolichospermum lemmermanii is one of the species we see, and it happens to sort of be buoyant with pine pollen. So we do see quite a bit of it commingling with the pine pollen in June, but it seems to persist throughout the summer and in a lot of our lakes, in the majority of our lakes. We're not sure how toxic it is, and so that's another element of the concern with cyanobacteria, is that these are these microscopic organisms. There's a whole bunch of different types. And they're not all toxic, but how do we know by looking at it? We really can't tell. And sometimes we can't even tell if it's cyanobacteria. This is Gliotrichia. It's a very common one that we see towards Labor Day in Winnipesaukee. In fact, it has just these little kind of dots that you can kind of see in the water. Um, it's just kind of a unique type, and we're studying it across New Hampshire. Nostoc is another type that you might see, not this like blue-green scum, but more of like a blue-green marble, something that's kind of intriguing and pretty. Can also live in Arctic lakes, and they can grow to be pretty large and almost opaque in color. So they're not just where things are warming, relative to the global warming conversation. They've been around for a long time, and they can grow in a lot of different environments and in different ways. In some countries, nostoc is considered a delicacy, something to consume. In other studies, it's found to cause neurological disease. Then we have some benthic mats, and this is also a bit unsightly, but just a natural part of the benthic community, especially in clear lakes where light can penetrate to the bottom. We have this like kind of rug effect of organisms at the bottom of the lake. And when it dislodges, it can come up to the surface. And then again, that surface accumulation can kind of get blown around. So our beaches usually get the unsightly, accumulation effect. But this is another type of cyanobacteria that we don't know a lot about in New Hampshire because we don't typically see it. It's hard to monitor when it's all the way at the bottom of the lake. We're also kind of seeing some kind of unique color blooms. Warnachinia is not a unique organism, but when it blooms, almost in a monoculture, as we saw in this pond. That color is truly like this yellow orange effect that obviously causes concern. People thought maybe paint, but a lot of people knew better that it was a bloom. So again, don't just, jump in that water. Kind of take your time looking at accumulations, because even if it's not a toxic cyanobacteria, we do find accumulation of a lot of microorganisms. I've been liking to say lately, if you can see a microorganism, there's probably too many of them. So this common summer problem can kill your dog in 30 to 60 minutes. It's a cute toxin. We get really upset thinking, well, we don't know how toxic it is. Are the fish dying from the bloom, from the lack of oxygen? Are dogs actually getting sick? This has been the biggest concern, I think, for acute toxicity, is that an animal or someone a small child, maybe even, who might swallow too much of a bloom, could get very sick. But it's very hard to track. And so there's lots of different routes of exposure to consider: ingestion, skin contact, inhalation, and eye contact. There's a whole bunch of different effects, affects people differently. And there's a lot we don't know about chronic effects. I won't get into this, but just to kind of boggle your mind that these are chemicals. These are secondary metabolites produced by these organisms, and they can produce them, shut them off, produce more than one type. They can also kind of turn them on and off and have these synergistic effects. And they all have very different effects: neurotoxic, hepatotoxic, cytotoxic, genotoxic, tumor promoter. We just don't know enough about how all this stuff. But I star the microcystin because that's the one that we have a few guidelines on in this country. Well, maybe not. And cylindrospermopsin as well. It's just been kind of a loose guideline on drinking recreation. But how toxic are these? We don't really see people dying. People are getting really upset about the message. Are these toxic? Well, they are, if you believe in LD50s. I mean, we're looking at, the comparison of other lethal organisms that produce toxins, such as the hook-nosed sea snake. It's on par with some of those. Of course, you're not being injected with, this stuff when you're swimming, but we know that it can be toxic, and there's a lot we don't know. In fact, in 2020, and I'm sorry to show this to you guys, and if you've seen it before, I apologize too. It's pretty gruesome, but in September of 2020, several elephants died from consuming toxic cyanobacteria. We didn't hear about it. It was a rough year, but this isn't just like, oh, maybe a few dogs get sick. It can really take down a large animal, and it's very concerning for the environment when we don't have fresh water available for anything. And of course, we're also still learning about the food web. And this is a study that took a very long time to come to the conclusion. I think it was published in 2021. However, they discovered a new toxin through this really unique food web, trying to figure out what was causing this kind of neurological disease with the bald eagles. And so they found a really interesting food web connection with the coots, who were eating hydrilla, who were harboring cyanobacteria. Came up with a whole new neurotoxin discovery in that. It took a long time to come to that. But just to show that there's a lot of interesting connections in our food web. Of course, we're worried about drinking it, and we're worried about our dogs. That's kind of more of the New Hampshire message, but we are also looking at a lot of aquatic food web studies. We've looked at loons, we're looking at plankton and some fish and some bivalves. So New Hampshire has its own concerns, with our lakes. And so New Hampshire DES, I think, is doing a great job. I would say a better job than I did when I was there. And they have come up with some really great ways of tracking blooms, reporting it, keeping your community informed, and subscribing to newsletters and news feeds on advisories for your lake or for several lakes. And so I just say, keep monitoring. I found this old posting, I don't want to advertise for it, from the summer: seeking water quality monitoring, alternates or volunteers. So, get involved, report things that seem suspicious, protect that watershed. And I had to end it there. I wanted to talk more, but I really want to hear from Jill. And so I will take questions at the end. Thank you guys for having me. I'm going to stop my share. Thank you.
Thank you, Amanda. So, like Amanda said, we'll get to questions at the tail end, and so we have enough time for Jill to present on eDNA. Jillian Emerson, my colleague at Green Mountain Conservation Group, earned her BS in microbiology and molecular biology with a minor in chemistry from Quinnipiac University in 2008. After her degree completion, she spent 10 years working in academic research in the Department of Molecular and Systems Biology at the Geisel School of Medicine at Dartmouth College. Most of her work has focused in the area of circadian rhythms using a model fungal organism, Neurospora crassa. In 2018, she earned her ASCP certification in molecular biology, and she's currently the water quality coordinator for Green Mountain Conservation Group in Effingham, New Hampshire. And most importantly, she loves dogs. So thank you so much. And Jill, you can take it away.
Thanks, Tara. That was a really good attempt at Neurospora crassa.
Thank you.
It's a very hard organism to say, and there's not many people in the world who study it. So that might be the only time you say that organism.
Probably.
All right, so let me see if I can get my screen shared. And all right. So hopefully everybody can see that. And as Tara mentioned, my name is Jill Emerson. And for those of you who heard the word Geisel and went, I think I know who that is, but I don't, that's Dr. Seuss. So I literally spent 10 years working at what we jokingly refer to as the Dr. Seuss School of Medicine, which I'm pretty sure the folks at Geisel did not like that we called it that. But that is what anybody who worked there did refer to it as. So as Tara mentioned, I am the water quality coordinator right now at Green Mountain Conservation Group. And I mean, Tara did a really good job of talking about me, but I do have a really quick slide about who I am, mostly just to show you goofy pictures during my various careers that I had. And I did spend 10 years doing molecular biology research in the genetics department that then got renamed before moving over to Green Mountain in 2018. And I'm really excited to talk to you guys tonight about our environmental DNA project. But before I get into that, just for those of you who are unfamiliar with Green Mountain Conservation Group, we are a watershed organization in Effingham, and we care deeply about the protection of natural resources with an emphasis on water. We are over the largest stratified drift aquifer in the state, and we do take its protection very seriously. So we have a lot of programs that are geared towards drinking water, but we clearly also monitor surface waters as well. And we have a very large community science program called Rivers that we do with our partners at the Saco River Corridor Commission in Maine, because our watershed is part of the greater Saco River watershed. So you all live in a watershed? Chocorua Lake is part of the Ossipee watershed, and here is a quick infographic on just the Ossipee watershed to give you a little bit of place. And like I said, that this is part of the greater Saco River watershed. The Ossipee is about half of that watershed. The Saco part, bigger part of it is in Maine, and it all flows out into the Saco and Biddeford area. So our organization believes in 4 pillars in order to protect our natural resources. So research, education, advocacy, and land trust, or the REAL principle. Clearly, I'm part of the research aspect of the organization, but I do also help out in the education area as well. And we do a lot of different projects at Green Mountain. So I am talking tonight about the eDNA, but as Amanda mentioned, we are also heavily involved in lake monitoring. We monitor 9 lakes over the summer, some of them monthly and some of them bi-weekly. So we are doing the Ossipee Lake system, so all of the bays off of that. We are helping to take care of White Pond and Ossipee, and Round and Loon Lake in the town of Freedom. And so here you can see a bunch of young folk out doing things. We are also an AmeriCorps program. So we have up to 4 AmeriCorps members who join us every year to learn about research and education and land trust initiatives, with the idea that they will learn some skills and then take it off to their next job and maybe bring a little bit of the Green Mountain way with them. And so it's really a good program to get their, literally and figuratively, their feet wet in terms of what it means to do research and participate in conservation. So environmental DNA, it's not necessarily a new idea as much as it's really been gaining traction in the last, 5 to 10 years or so. But basically, environmental DNA is just DNA you harvest from the environment. And so many of us who enjoy the outdoors clearly follow the um, leave no trace mentality, right? We take only pictures, we leave only footprints, and that's that. Despite your best efforts, you are actually, though, still leaving bits and pieces of you behind, probably in skin cells, maybe some hair, and for those of you who are extremely unfortunate on the trails, maybe a little bit of blood. And so all of that is DNA that you are leaving behind that can be utilized for a biomonitoring or a conservation management purpose. And so there are many different applications, and I feel like every day there's a couple more papers about people using environmental DNA in cool and interesting ways. But we're using it in a couple of specific ways here at Green Mountain. And so the first way that we are using environmental DNA is we're using it to look for invasives. We have a lot of invasives that are threatening New Hampshire. Some are already here, and some are in the state surrounding us, and so we know that they are coming. And so utilizing environmental DNA has the potential to give us a little bit of a jump to help protect some of these lakes. If we can find these organisms quickly, we may be able to minimize the damage that they can do. We're also using it to look for species of conservation interest. So kind of the flip side of that coin is we have organisms that we deeply care about in our state. A lot of us are involved in, brook trout restoration projects. So brook trout is one of the species that we can go out and look for, but there's a lot of different types of species of interest in New Hampshire that we can work to develop systems of monitoring for. And then the second way that we are using environmental DNA is for fecal contamination issues. So as our recreation increases in our state, particularly on our freshwater areas, we're finding that there are higher E. coli events. And so when we have those high E. coli events, we really want to try to start figuring out why we're having them and where the sources are coming from, and whether it's a human source or maybe something like a geese or dog or deer source. And the way that you would solve for those problems are different. So having a little more information about fecal contamination could be really useful for conservation management. So what I'm going to talk to you tonight about is about the technique called DNA barcoding. So there's two different main ways that you can look at environmental DNA. So you go out into whatever environment that you are interested in looking at, you collect your sample, and in your sample, you're going to have a variety of DNA from whatever organisms recently were around. The first method, which is the one that Green Mountain is using, is called DNA barcoding. So basically what we're going to ask is, hey, I want to know if this one organism or these couple of organisms were in our DNA pool. And so then we can extract them, we can look for that DNA specifically to identify that organism. The other method is called DNA metabarcoding, which instead of asking, hey, is this one organism here? What you do instead is go, hey, what organisms were here? So you're getting a more complete list of all of the things that were there at one time, probably within the last day or two. And so it's a little bit more of a complete record. It is a little more complicated and a little more expensive, as you can imagine, to do that. And since this was our first foray into the world of environmental DNA, we decided to do the DNA barcoding technique. So what is DNA barcoding? I keep using that term. Well, if you've ever gone to a grocery store and you've scanned a package of something, scanning the barcode, and the machine beeps at you that you just bought a 32 pack of Oreo. Well, how did that machine know that that was a 32 pack of Oreo? Well, obviously the barcode. So the barcode has different line thicknesses. It might have a different number of lines. It's got some numbers associated, but that barcode is unique to that item that you just bought. So we in genetics have that same type of barcoding region, and there's a couple of different places that we can target that are very unique to each individual organism. Um, and so one of those places where we have unique sequences is actually in the mitochondria. And I'm going to ask you guys to, think back to your freshman biology classes, which was probably the last time you ever thought about a mitochondria. And your one factoid for bar trivia night might be that the mitochondria is the powerhouse of the cell, and that is about your extent of knowledge of the mitochondria. The mitochondria actually has its own DNA associated with it, and that DNA is really unique. We have something in humans called mitochondrial Eve, so all of our mitochondria comes from our maternal line, and you can trace it back throughout humanity to one person in southern Africa, however many hundreds of thousands of years ago, millions of years ago. Mitochondrial DNA, there's actually a lot of it. There's more of that than there is nucleic DNA. So it's a good source to target because it tends to be pretty robust. And we look at one very specific region called the COI region. So when we're doing these screens on animals, we're utilizing that mitochondrial DNA to try to find this really unique sequence to figure out what animal it's from. If we want to do this with bacteria, now we're looking at a ribosome. So the ribosome area is where you make proteins. And it's an area also where if there's ever any sort of mutation that happened in there, the organism would die because you can't create proteins. So these areas are highly conserved. Same thing for plants. If you're going to do a survey where you're going to target plants, you look at chloroplasts because they too have their own DNA. And all of these things, what they have in common basically is that if there are mutations in our mitochondria or ribosomes, or for plants to chloroplasts, they're not going to survive. So these areas are highly conserved, meaning that they don't really mutate. And then for the work that I did in fungal organisms, we're back to that ribosome. So ribosomes tend to be one of those really targeted areas. So once we found our special area that we want to look at, you have such a small amount of DNA in your big pool that you need to create a little bit more of it in order to do anything. So we move on to this technology called polymerase chain reaction. So basically, starting up in the upper left-hand corner here, you see that the blue is your DNA sequence of interest. So let's say that that's the mitochondrial DNA of a human, and that's what we want to look for in our big pool of a whole bunch of other DNAs. Well, we can create these like special DNA sequences that literally will do a search and find. They're going to look for that special DNA sequence, that human DNA, and they're going to bind to it, and then you're going to make more of it. And so every time you go through a cycle of this process, you double the amount of DNA that you have. So if you had one strand to start with, by the end of the first go around, you've got 2, then 4, then 8, then so on, until you've made thousands or millions of copies. So this process is basically just taking your very small, very finite amount of DNA and creating a lot more of it in order for it to have it be usable, in order for us to see it. So once we've created all of this DNA, now we're going to run it on a gel using something called gel electrophoresis. And so the next couple of images are going to be a lot of gel images. So I wanted to just go quickly through how do we look at a gel image. So what we can do with the DNA that we just amplified, we can run it on gel in order to visually observe it. So you load your DNA at the top. DNA is negatively charged, so then we put an electrical current through there where we have the positive electrode being down south. So basically, you're running from negative to positive. Your DNA will actually move through the gel and separate itself out by size. So the smaller little pieces that we're looking for will go faster through the gel, and the bigger pieces kind of get clogged at the top and they take a longer time to go through. Every DNA gel electrophoresis has a ladder associated with it, so we can know the size of our DNA. So the cartoon on the left is kind of giving you a little like easy snapshot of what a gel would look like. And then on the right is actually one of my images where we were looking for Asian clams. And so we made our little specialized sequences of DNA that were very specific to Asian clam. And we went, hey, in this system, can you find Asian clam? And in here, this was a control that we did where we visually observed Asian clam in the sample, but our system was also able to confirm, yes, we did have Asian clam. So once we designed all these little special sequences of DNA, we needed to actually try it to make sure that it worked. Because as many of us know, sometimes things look good on paper, but then when you actually go out into the real world to use it, it doesn't quite work. So over the summer, I took my AmeriCorps team to a couple of different locations where we had either known organisms of conservation interest were there where we could visually observe them, or conversely, places where we knew that there were infestations of invasives. Um, so here is our AmeriCorps team going out and collecting some water samples at various depths for some of these organisms. When we're looking for these organisms using environmental DNA, we do need to be making some educated guesses in terms of we want to collect samples where we think the organism would be. So environmental DNA does not really work if you just stomp out on your dock, you collect a scoop of water, and you're hoping that you're going to find some salmon that may be in the much deeper, cooler water, 100 yards away. So we went to locations where we knew these organisms would be. In cases like spiny water flea, we were able to visually confirm them. And then we brought back the water samples and went, okay, can our system now find these things that we went out and we looked for? So here are our samples for some of the organisms of interest. So we partnered with the Squam Lakes Association, who had a interest in knowing what their salmon populations were like. They get a lot of requests from fisher people who want to know, hey, is there still salmon in the lake? So we were able to do a dry run on some salmon. And confirm that our system can, in fact, find salmon in a water sample. Same thing with brook trout. Brook trout, again, very popular in New Hampshire. We went to a hatchery where we were able to visually see that there were fish. We collected some water and we ran through our process and were able to confirm that there was brook trout. And I get same thing with spiny water flea, which is a new plankton invasive that is established now in Winnipesaukee and Winnisquam, but it is traveling. And so where we are a high tourist area with Lake Ossipee being the 6th largest lake in the state, we're expecting that we're going to see spiny water flea in our area soon. So we're working up a spiny water flea detection protocol. The next step was to then go out and try to use our system essentially like in real time, go out to a place where we are pretty certain we'll find some salmon in the wild, but we're not going to be able to now visually observe them. We're not expecting to find brook trout, but we're still going to run our tests just to see. And then we also ran some spiny water flea samples out on Squam Lake in the Broads region, for those of you who are familiar with Squam Lake. So we were again collecting at a couple of different depths where we thought we would find salmon, and we collected at some different depths where we thought we would find spiny water flea. And so the figure up above, you see that there's S and B on the bottom. So all the S's are where we tested for salmon, all the B's are where we tested for brook trout. And so in every salmon lane, you can see that there's this green band product. And so that's the DNA that we were looking for. So in this case, our system confirmed there was salmon in Squam Lake. The bottom green figure down there was for our spiny water flea. And so while we did do a visual survey for spiny water flea while we were out there, we again tested our system. We did not visually observe it, and our system also did not find that there was spiny water flea in Squam Lake. And as of right now, there has still been no reports of a spiny water flea infestation. So we felt pretty confident that our system was correct and that there was no spiny water flea here. And the goal of this is to try to launch it wider the next summer. We spent a lot of time learning what not to do this past summer. As anybody who's ever run a research project will tell you, that's kind of par for the course. And so we're hoping that we can go to more places and do some more trial runs where we're a little more in the unknown territory. So I'm very excited to be working on that this summer with our AmeriCorps crew. So secondly, the other way that we're using environmental DNA that I want to mention really quickly is in that fecal tracking that I mentioned earlier. A couple of years ago, Green Mountain started its own E. coli monitoring program, and I naively thought at the time that people would care about having high E. coli results. Come to find out when we were telling different lake communities that like, hey, there's a lot of E. coli here, they didn't seem to care as much as I thought that they would. So we sort of brainstormed about how could we get people to care a little bit more about E. coli, because E. coli can have very negative health effects on people, and we're trying to protect public health here. And so one of the things that we realized that if we told people when they have a high E. coli event, if we could say, hey, you're swimming in human poop, weirdly, people care about that way more than if you just say, hey, you're swimming in a high E. coli place. So using the same sort of principles of how can we find if this high E. coli came from humans, we can look for human gut bacteria that we're not necessarily finding out in the wild on its own unless it's coming from a fecal source. So we targeted a couple of different organisms in that case. So a lot of these are just various gut bacterias, but we also targeted a double-stranded DNA virus called the John Cunningham virus, which 94% of the human population has been affected with. Typically by the age of 4, it was some kitty crud you probably had that, got sort of hand waved away. But we shed that virus now for the rest of our lives. So we're basically using the same thing. We're tapping into very specific organisms that have co-evolved into humans and seeing if we can find it in our water samples. So there's a picture of Grace on the right there, processing some of our E. coli samples with her cone of sterility, which is the flame that we use, which is a lot of fun for the AmeriCorps members to essentially play with fire for a few minutes. So these gels are very confusing, but I did think that this was a good example of how our protocols can evolve over time to be more efficient and a lot cleaner. So this was, again, using a known sample of human sewage. And can we find all those different gut bacterias and those viruses? And the answer was yes, we could, but our systems needed a little bit of cleanup work. So here you can see we can find all of those different human bacterioids and JCVs, the John Cunningham virus. But it's messy. There's a lot of bands. It's not one nice clean one. And then over time, we're able to evolve those PCR reactions to get us one clean band in our system. So basically, we were able to see, yes, we can find these gut bacteria in a known sewage sample. Now let's see if we can go out in a high E. coli setting, and can we still find these organisms or not? So this was taken in a place in Maine that is near that is next to an area of high E. coli that has pretty traditional high E. coli values in the summer when we test there. And so we took a secondary water sample and ran our system through. This one was not quite as conclusive as I would have liked. We did come up with some human markers, which you can see with the green check marks that those are humans. We also did a series of goose markers because the next likely organism in our watershed, if it's not going to be human poop, it's goose poop. So we didn't come up with really any goose hits, but we would have liked to have seen a few more human markers in this. But these trial runs really taught us a lot again, a lot of the summer was spent figuring out how not to do something in order to figure out how to do it correctly. And so thank you very much for letting me talk to you about environmental DNA. There's a lot of other applications that this technology can have, and I'm really excited that Green Mountain has allowed me to bring this technology in and to start utilizing it in a couple of really unique ways. And thank you very much for your time.
Okay, we're going to open it up for question and answer. I think there are only a couple in the chat. Let's see, I'll start out here. There's one from David for Amanda. Are there specific temperature ranges that favor these cyanobacteria? Are there more blooms in warmer conditions?
Yeah, I think we see cyanobacteria are able to survive all year. There's not a temperature that really kills them. In fact, if it gets too hot, they're not too happy in culture, so 70 degrees, everyone likes 70 degrees. I think that's usually when we start to see them really take off. But in fact, we're seeing a peak in August in New Hampshire, so you can imagine it really does kind of follow that temperature gradient of what we see in New Hampshire with abundance. But we're not seeing a ton of it. It's just it really kind of comes with the waves of rain followed by like calm, stagnant days of hot weather. So there's sometimes a more complex factor behind it than just the temperature. But typically, globally, yes, they like it hot. They like it warmer.
And we're going to see more of it as waters warm. I hope that answers it. It's a little complex. It really depends on the organism, because they're as different as plants.
Okay, and David follows up. I think you kind of touched on this already, but Amanda, are there particular conditions of lakes that allow or prevent toxic algae? What can be done?
Yeah, it's really hard to know how toxic the conditions are. But if you can see it, it's just something to report. We are trying to test, and that's where, you know, some of the methods that Jill's bringing up could be useful for us in the future as well, where we could develop primers for specific water bodies. We're working with the town of Wolfboro, Bigelow. I can see Larry's here, Mirror Lake. There's a lot of groups involved where we are trying to evolve and look at some PCR methods for determining toxic cyanobacteria. However, we're not finding very good patterns. We find that it really depends on the species. And if we're only concerned about microcystin, then maybe we should be really primarily concerned about seeing microcystis, which is one particular type of cyanobacteria. I wish it was more straightforward. You know, I think that this idea of toxicity is really under researched to some extent, but also just very complex. We're talking about one toxin, where there's so many different types, and some are liver toxins, some are neurotoxins, so they just behave differently. So it's really important to know what's growing and what we have, and what might be promoting their toxicity. And we have seen correlations with higher nitrogen levels promoting toxicity, some iron reactions that can also control genetics. So it's not so straightforward. And in fact, you know, we talk about the health food supplements and the things you can get a whole scoop of spirulina and add it to your smoothie. I mean, that's cyanobacteria. Those are not necessarily being tested for all the toxins, and we're discovering toxins quite frequently, in fact. So I think it's just I think the when in doubt stay out method is important to consider. But again, it's, you know, how toxic is it, how bad is it? It's like kind of all relative at this point. I again hope I answered that question without too much.
All right, so Todd is asking about cyanobacteria. Is it an algae, a plant, or living creature?
Okay, yeah, I mean, it's living. It's been around for a long time, but it's a prokaryote, so it's not truly a plant. I mean, we can call it algae, but it's a photosynthetic bacteria, so it can really outcompete other algae. It can divide and conquer pretty quickly. It can grow pretty rapidly. And some of them have specialized cells that allow them to kind of outcompete and use other types of nutrients from the atmosphere, convert what they need pretty quickly. So they're pretty unique in their own way. They're considered phytoplankton. I guess they're just not truly algae, but you know, we can, I think it's still important to recognize them as blue-green algae, because all of the supplements will say blue-green algae added for health effects. And it's important to remember that that's the same thing, but it's truly photosynthetic bacteria.
Okay. Juno wants to follow up on cyanobacteria coming after days of rain and then stagnant warm days. Is that because the rain is washing nutrients / food into the lakes?
Yeah, exactly. And we see that pattern with certain lakes. And then other lakes, you know, they might say, well, you know, actually without all that runoff, those hot stagnant days, in our runoff, and I'm sorry, in our I'm trying to say, you know, our outlet, our flow, our retention time is quick. It can vary depending on the system. So I think typically at beaches, we were starting to see that pattern though of like, uh-oh, we got a lot of rain. It came down quickly. And now it's a weekend of hot sunny weather. And we just knew it every time that we were going to get more calls. I wouldn't say I would be able to predict with certainty, you know, which lakes. It was sometimes a surprise, but there are some lakes that are experiencing patterns and habitual issues.
Okay. Ed is asking, Silver Lake had a cyanobacteria warning last summer, he thinks. Would that be more on the southern end of the lake where the wind would build it up? We swim on the northern end.
Was it Hollis? It wasn't Madison, was it?
Madison, Silver,
Really?
I'm guessing Madison because Ed, yeah, I know
Yeah.
Ed.
I'm not familiar with that bloom if that happened, and I could follow up with you if you want to email me. But yeah, we see that, Silver Lake and Hollis, for example, the state park, gets the wind kind of, it's a very small, shallow system, but if you go out to the middle of the lake, it looks pretty clear. And it just, all the blooms just keep piling up. It has more of that like long pond kind of shape to it as well. So you will see wind either actually helping it disperse and kind of mix where you don't see it, or kind of pushing it. So there can be upwelling, and there's a lot of kind of crazy factors of it, with the if you've ever even just like thrown something in the water and see which way it goes, we've tested like every depth can have a different direction, so it can be complicated. But I'm sure if you're seeing it particularly building up in an area like a swim area, you might realize that's going to be the prevailing location where you find it.
Okay. Bruce is asking, in a body of water with a strong inlet and outlet, would higher dissolved oxygen reduce photosynthetic bacteria?
Yeah, it's interesting. Again, we do find different types. So in some of these like really big clear lakes where we don't see a lot of organisms at all, we might find more of the pico cyanobacteria, which we don't know a whole bunch about. But we are finding that lakes that have more oxygen are going to have better balance of organisms. What happens with these stratified lakes in the anoxia, and then that pattern of blooms kind of dying and decaying, going down to the bottom of the lake, is typically more of an internal loading kind of vicious cycle of low oxygen releasing more nutrients that have just been kind of down there waiting. A bloom happens, it dies, it goes back down, it creates more of an anoxic situation. So it can kind of trigger a bad cycle. But again, I mean, so yeah, a high flow, high turnover, and plenty of oxygen is what we typically like to see in our water bodies.
All right, so Bob has a question for Jill. Hey, Bob. What is the lowest E. coli concentration that you can do eDNA analysis?
So in a very unsatisfactory answer of it depends. So which I feel like is kind of the broad strokes for science of every situation is unique. Typically for a standard PCR reaction, you need between like 25 to 100 nanograms, but in a lot of cases, it's not. The quantity does matter, but it's the quality that matters more. So if you have like really degraded, cruddy DNA that has a lot of proteins or RNases that are still DNases that are still attached to it, that's not going to amplify very well versus if you've got something that's a lot cleaner but maybe a lesser amount, that probably would PCR up just fine. It also depends on the type of DNA that you're using. So things that are more that are smaller and more purified, like when you do work with plasmids, you don't need as much of the plasmid as you would for like whole genomic DNA because you've got so much more sequence in there that you frankly don't really care about because you're looking for one tiny piece. So it's more on like the type of DNA and the quality, and then the third thing is the quantity of DNA for the reaction.
Okay, back to Amanda for this one. What are your thoughts on alum as a treatment for cyanobacteria?
Oh, goodness. Well, you know, I was the Lakes Lake monitoring program was lucky. We got to kind of follow the recent development with Lake Canasaka, if you're following that. So we've been monitoring that pretty carefully with the state and with a consulting firm and the volunteers. I don't really know a lot about how this will go. I think it works. It seems to be a sufficient treatment for some lakes and not for others. It's definitely not for every lake. And I think it takes quite a bit of thought and planning to decide to get to that point. Because that's obviously not where we want to be. And for that matter, I like, you know, I can't really say how I feel about, you know, algicide either. It's one of these topics that we need to learn more. We need to monitor. We really need to be careful in the application. And we also have to have a good plan for it. So it's not one of those things that you can just, you know, eventually buy over the counter and say, well, we're going to do this. Obviously. And I hope that doesn't go that way with algicide and other ways. We are unfortunately hearing people are trying to take matters in their own hands with some stuff. And, you know, this alum treatment was a very carefully planned, project that, you know, hopefully will last as long as they think it will. It's expected to last probably 10 to 15 years as far as keeping nutrients low, as long as everyone does their part in the watershed as well. So it's, you know, it's a compounding issue. So if anyone wants to email me more about that, I could talk separately about that too.
Okay, thanks, Amanda. We have a couple of questions for Jill from David and then Deb. They want to ask you directly. So go ahead, David.
Thank you very much. You guys, it is so wonderful that you bring such depth of knowledge to this conversation. Really, really appreciate it. Jill, are you working with Deb Marnich on the eDNA analysis of our proposed fish ladder or aquatic organism passage at the dam in Lake Chocorua?
So Deb has talked to me about this last summer. I'm sorry, time has lost meaning since COVID. And it is something that I would definitely like to be involved in. But I do know that she was working with Allison Watts from UNH. And I don't want to step on toes, but I would love to be certainly a part of that and to see if there are ways that we can work together. We're still very much piloting our system, and the more opportunities I have to get out and try it, the better we're going to make our system essentially. So I would love to be involved. Hi Deb, I would love to be involved.
I think it's meta analysis that we're looking for, and I should be quiet because I am not the expert. Deb is doing a great job setting this up for us. So
thanks. I think that's my intro. Thanks, David. And Jill, thank you so much for your presentation. And Amanda, thanks for your presentation as well tonight. It's been truly enlightening. And as Jill mentioned, we did talk last summer about eDNA analysis. The reason being we are rehabbing a small dam that we have on Little Lake, and to enable fish passage, aquatic organism passage, we are adding on a rock ramp that will facilitate that. In the process of creating the rock ramp and habitat connectivity, we thought it would be best to sample eDNA for conservation management for habitat connectivity. And we are testing for bridled chiner, brook trout, and slimy sculpin, species that are found in a cold water system but may migrate at certain times as well for reproduction at certain periods in their life. We may also electrofish to test for them. However, we are just dipping our toes into this data analysis. We started in November, collected our first samples. And as Jill mentioned, we're working with Dr. Allison Watts from UNH, and we are doing DNA barcoding actually. So we're looking for one specific fish or a few. So our analysis is forthcoming, maybe in a few months. We haven't gotten results. We're hoping to collect some baseline data and compare the data of what's there now to what will be there once the fish ramp and the aquatic organism passage has been restored. So again, we're using the data collection for conservation management purposes. And it's been great and really enlightening to talk to Jill about this. And I hope we get a chance to partner in the future on a project like this. Thanks so much.
All right, so moving on, we have a question from Rich. Can the DNA testing distinguish between wild native trout and the hatchery reared genetically identical farm raised or reared trout? Stocked trout for angling versus wild trout, or is the DNA just able to ID brook trout?
Um, that's a good question. There is maybe a way to do that, but it would require a lot of sequencing, like whole genomic sequencing to look for variations between the two types of organisms. So it would be complicated to do, and it may ultimately not be successful, depending on how much genetic diversity there are between the hatchery raised and the wild native ones. Um, that COI region is probably identical between the two of them because that is more for like a species determination, but there are probably some genetic differences elsewhere that one could look for. It would take a lot of sequencing to do that because you couldn't just take one wild trout and say that it is representative of all of them. You would need to get probably a reasonable number of samples in order to do that. So there could be a way to do it, but it would be probably more complicated than what it's worth, to be quite honest.
All right, so we have a question from Bruce. Given the amount of eDNA needed to identify E. coli, are standard chemical tests for nitrates, nitrites a better way to identify lakeside septic failures?
So when we're doing our fecal tracking, we're using the high E. coli as an indicator that there is fecal pollution, but it's not telling us if that's a human source or a goose source. So if you're looking just for like at nitrates and phosphorus, you're going to find that in fecal material regardless of the organism because that's just part of fecal material. So if you're just trying to test for pollutants, yeah, you can certainly test for nitrates and phosphorus. But the issue that we were having when we were telling people like, hey, there's really high E. coli here, is that like now we don't really care. Versus if they had cared to begin with, then they would already be doing those testing to see where it's coming from. It's trying to show that the source is human versus not human, which nitrogen and phosphorus is not going to tell you that. That's just going to tell you it's a fecal contamination.
Okay. Rich says, thank you, Jill. And Juno is asking, this one's for Amanda and Jill too, because I know you have experience in this. Can you make recommendations for what folks who care for lakes, or live on or near lakes can do or not do to avoid feeding cyanobacteria blooms?
Yeah, well, yeah. I would say there's a lot we could do. We're talking a lot about best management practices. Sorry, I kind of got off offhand on the feeding because I was thinking, well, don't feed the birds. Don't, that it's all the same things that we're trying to protect the water quality. There's lots of little things we could all do. But reducing that stormwater effect is really important. We're seeing huge increases in chloride as well to indicate that nutrients are getting flushed in very quickly with salt. And those things can cause adverse effects, can quickly feed a bloom. But there's like a lot of things like reading those signs, don't feed the ducks, don't feed the geese, clean out your septic system, identify areas of runoff. There's a lot of community could do to get those best management practices together. And I also recommend New Hampshire Lakes has some great resources as well, where you can become lake smart. New Hampshire's got a lot of great resources to help you along to identify those issues. So please reach out if you want more information. Jill's got a lot of good ones too, I'm sure.
Yeah, just to sort of piggyback on Amanda, I'll put a plug in for Soak Up the Rain, because they're a really good organization for all of you DIYers out there who like to sort of do your own and don't want to pay for a landscaper to come through and do something. There's a lot of how-to guides for how to build a rain garden, how to put in a swale, how to put in a water bar. Friends of Danforth Pond too recently just worked on a 319 watershed assistance grant in our general area where they installed a lot of different best management practices. And I know that they would be thrilled to show them off to other lake communities. And so if you are interested in like learning more about the nuts and bolts of like how to install a water bar, we can definitely put you in contact with that group over there, because they created a very detailed set of like blueprints and materials lists out there. But yeah, just anytime when it rains, go outside and see where the water is going on your property. If you live near a lake or a river, and if the rain is just going unimpeded right into the water, then that's a problem. And that's where you could be installing a BMP. So that's my recommendation for landowners. And I weirdly get a lot of people who comment that they throw their grass clippings in the lakes. So if you've got a neighbor who's doing that, have them stop doing that.
Definitely. And maintain those riparian buffers. Vegetative buffers is my plug. Let's see. Do we have any more? We have lots of great links in the chat. If folks haven't seen those yet, Juno's been doing a great job of adding things, like New Hampshire Lakes and the Lake Smart program. And she also echoes riparian buffers. Absolutely. So that looks like that's all we have for questions, unless anyone wants to jump in and ask a question live before we sign off tonight. And thank you both to Jill and to Amanda and to Mary for helping us to put this program on tonight, and Juno. We have one more comment here. Thanks for the informative talk from Benedict.
Hello, Larry Gill. Can you hear me?
Yes.
Okay. Question about should we be trying to link stream influences and cyanobacteria? What I mean is that we have a single source primarily as inflow. And we do have some information about that stream, but I don't believe we have anything that indicates that there are cyanobacteria within that stream, that the cyanobacteria, and probably there are from the past history, but as a causative factor, or is it more than nutrients?
You mean like it would kind of flow in and then now
Yes.
it's here? That
Yep.
that can happen. It can. We've seen it happen, but more on an ephemeral base. I think the conditions of the lake really need to be there for the cyanobacteria to grow well. They seasonally change as well, but you could certainly see something come in from a stream. They don't typically do well just growing in streams because of the nature of it, high flow, high oxygen. It just keeps moving, but it can certainly pass from lake to lake. We have seen that, but it's not typical. We don't really see blooms that large that would get through to that extent. So both, but it is worth monitoring those streams, especially for tracking everything, nutrients and what might just be a problem upstream that's causing big problems downstream.
Okay. I guess one more question about pico cyanobacteria. You mentioned that
Yes.
as an area that you're interested in.
Yes.
Could you speak a little bit about that?
Yeah, absolutely. There's just so many different types of cyanobacteria, and I took an interest to the picocyanos, which are so small that we under the microscope, we just don't really know what they are. We just look at them as little dots. And so that would help to employ this eDNA method, which we have been doing with communities like Jill's group, like your group, and Mirror Lake and Wolfboro as well. But these picos we don't know a lot about, but they're probably the most numerous organism on Earth. They're in the oceans, they're in the lakes, and we're finding them in the air. And so we're just trying to learn more about them and how they are incorporated into the food web. We don't know how toxic they could be, but more so they're just like a whole different category just because of their very, very small size. Anyway, yeah, there's a lot we don't know about cyanobacteria. So those are one of them. That's a little mysterious, but they prevail in our lakes.
Another quick question for Jill, I think. Aerosols. Do you have a way of sampling for aerosols? Because I know that Dartmouth had looked at that issue as a cause for cancers, I believe, brain cancers, and so the chronic exposure. Is there a way, or do you know of a way of being able to sample aerosols?
So I know what you're talking about is Lake Mascoma in the Dartmouth area, because that's been one of the little hubs for research for, I think, yeah, both cancer and ASL, because Lake Mascoma for 20 years or so has had issues with cyanobacteria. I don't have a way, I don't do anything with aerosols personally. Amanda, I'm not sure if you can.
Yeah, I'm like, I should just.
Yeah, I was gonna say you're doing more of that
Yeah,
than I am.
Mary, I could follow up with you, but UNH is doing aerosol studies. We did do that study on Mascoma Lake. In a former life, I did that with Jim Haney, as my name before Amanda Murby. So I can talk a lot about that Mascoma study if anyone wants to another time. We can test aerosols. We just don't really know what we're finding. There is an aerosol effect. And what we're very interested in is the non-wind generated one, because a lot of studies are looking at sprays. And, if you go skiing, water skiing, or you get, tubing, you get a lot of water in the face, but there's an evaporative process that also occurs. When that fog lifts off the lake, what is that holding? We are seeing a lot of pico-sized organisms. I'll follow up with you, Larry.
Okay, thank you.
Okay, last chance. Any other questions? I have a few messages to pass along from Juno. Anyone else have a question for our speakers? Well, great questions. Thank you for all the questions, and thank you, Amanda and Jill, for being on the cutting edge of a lot of this research and doing so much for our New Hampshire lakes and watersheds. Juno